Hi, everyone. Nice to be here, even though I can't see anyone. I trust some people are out there. So I've been coming to the Tucson conferences for about starting 18 years ago, and they've been a very foundational part of my both academic and personal development. And I'm very happy to be a part of it now. I'm coming from neuroscience, and I'm really interested in information as it manifests physically and what can be said about the contents of inner experience in connection to what we can see physically. And really many of the topics at the conference are really appealing to me. I'm currently an assistant professor at the Claremont Colleges, which is a liberal arts school where we try to bridge, kind of like this conference, we try to bridge different disciplines from science and art and humanities and ethics and try to get kind of an interdisciplinary conversation going around this. So I think that's all I'll say with respect to an introduction, and looking forward to hearing your thoughts. So today I'll be talking about work that I've done with my former advisor, Fritz Sommer. And it's using data collected by the lab of Albert Lee by Brian Lustig over there in Genelia Farm. So when you stick an electrode into the brain, you see different kinds of signals depending on where you put the electrode. So at the finest scale, if you're right next to a neuron, you can see things called action potentials. And as you pull your electrode further and further out, you start seeing local field potentials, which are the collective activity of hundreds of thousands of neurons. And even further out, you start seeing electroencephalograms, EEGs, collective activity of millions of neurons. And so there's a trade-off, because the deeper you get, the finer your resolution is, but also it's harder to access that tissue. So one of my dreams is to be able to read out the contents of inner experience non-invasively. And there's a lot of precedent in this. So for example, if you look at the EEG of a person, depending on the state of mind they're in, you see very different signals. Depending on, for example, if they're excited, relaxed, drowsy, etc. And one region that I've been focusing my work on is the hippocampus, and it's home to a very prominent brainwave called the theta rhythm. It's about an 8 hertz rhythm, and it's studied intensively in many species, especially in rodents. And this rhythm, while it's recorded in the hippocampus, it's a global rhythm. It broadcasts everywhere. You can detect it in frontal cortex, visual cortex, among many other regions. And it also has the state-dependent quality. So that as an animal is still, the LFP, the local field potential of the hippocampus, looks very jittery and kind of sporadic. And as the animal starts running, it all of a sudden becomes much more periodic. And then you start seeing this 8 hertz beat, which is the theta rhythm. And now as the animal runs through space, we can start seeing the function of the hippocampus. So not only is the hippocampus implicated in memory, it's also implicated in navigation, going through space. And in the case of a rat moving through its environment, depending on where it's at, different cells called place cells will activate. So as the animal runs over here through the place field of this cell will start firing action potentials. And what's really neat is that the action potentials fall at very particular phases of the theta rhythm. So it's like there's some sort of synergy between the single neuron and these waves, to the extent that when you have many neurons that are firing at different locations of the environment, the theta rhythm can kind of chunk them into brief sequences of activity, performing some sort of binding of the animal's experience as it unfolds through time. And so this is kind of a prototypic example of a code that's not completely in the neurons, not completely in the oscillations, but somewhere in between. Now, one thing that we found really fascinating about this code, about the theta rhythm, is that it's very complicated. So when you don't look at the theta rhythm, not at one location, but at many different locations in space, it actually, you find many different rhythms depending on where the electrode is. And you can visualize this as a movie. So here on the XY coordinates, you have an anatomical cross-section of the hippocampus. And in Z, you have the voltage as being measured at each position of the hippocampus. And what you see are these beautiful traveling waves of potential in the local field potential. And depending on where the animal is in its environment, these waves change their shape. They travel in different ways. And whenever you have such complexity, it raises the question, what information do these waves hold? And that's kind of where we embarked on our project, trying to decipher the forms within these waves. And so we took inspiration from radio communication. So also in radio, you can have these oscillatory signals that might change different properties. For example, the amplitude of the signal or the phase of the signal, the timing of the signal. This might be something that you pick up on a radio. Now, to disentangle the message that the signal contains, you can use a trick from signal processing. You can convert the signal to what's called a complex valued signal. So instead of a sine wave, it becomes kind of a corkscrew shape, which is more amenable to mathematical analysis, to extracting these changes in amplitude and phase. And by applying a technique called demodulation, you can separate out what's called the carrier wave, this reference clock, so to speak, from the signal that is more slowly changing and has the content that you're hoping to recover. So now we want to apply this tool to real data. So here you have an animal, a recording from an animal as it runs across space, recording the electrical activity at many different hippocampal locations. And we apply some, the first thing we have to do is find the carrier. We don't have this very metronome-like carrier like the radio. So what we do instead is we use a technique called PCA, Principal Component Analysis, and this pulls out the loudest, the strongest rhythm component that's shared across these electrodes. Essentially, you can think of it as an average of some sort. And this we use as our carrier, as our reference. So now, when we subtract the phase of the signal at each electrode from that of our carrier, what we're left with is this very smoothly changing signal, which changes on behavioral timescales. We've kind of decoupled the intrinsic dynamics of the brain from these messages, which presumably convey something about these slower changes in the animal's behavior. And then what we can do is use a technique called linear regression, where we look for patterns across these spatial patterns across this demodulated signal and use it to predict where the animal is in space. So each decoder gets activated when the animal is at a particular location. And by looking at the most active decoder, you can say, oh, the animal is over here in its physical environment. So now we want to quantify how much information does the brainwave carry? Do these brainwaves carry about the animal's current state? And we do that using a decoder and comparing it to the decoding of spikes. And as I said at the beginning of my talk, neurons are thought to be the most precise carriers of the information of a brain region. So what we can do is look at the animal's instantaneous location on a track as it runs back and forth. Here I'm showing that in time as the zigzagging black line. And these red dots are an estimate of where the animal is based on the spiking activity of its single neurons. And now we want to ask, how well can we reconstruct the animal's position, not from the spikes, but from these patterns across these brainwaves? And the answer is that we can do it really well, even better than we can reconstruct the animal's position by looking at the spikes. But there was one problem in our original work, which is that our decoder of brainwaves didn't always work. Specifically, when you looked at the animal's position in a more complicated environment, where it could move any which way it pleased, the spikes did a much better job of tracing the animal's actual position than the brainwaves did. And we wanted to know why. And we had a guess, and our guess was that it had to do with the fact that the theta rhythm is state dependent. We're using this kind of oscillatory reasoning in our decoder, and we know that oscillations aren't really strong when the animal is still. And so that stands to reason that we can't decode something where there is no rhythm. So we wanted to dig into this further to see if we could do anything about this. And so we looked at a different task. And this is a task where our collaborators collected data with animals that spent extensive periods of time at the end of the track drinking water. So we have lots of data from these periods where the animal is still, which is very useful for developing better learning algorithms. And we wanted to confirm this idea of the rhythmicity breaking down during these periods of stillness. And that's indeed what we see. So while the animal is running, if you look at PCA and you use it to extract the carrier, you have the strong and rhythmic theta rhythm. And as soon as the animal stops at the end of the track, the rhythm seems to be there, but it's much more sporadic. And this can be easily seen in a spectrogram, where you have a very strong rhythmic component at 8 hertz. So this is a frequency time plot. And that rhythm, you can see it's still there, but it becomes kind of weaker and it starts drifting in frequency. So there's some sort of breakdown in rhythmicity. And now we can see how much information these waves carry about the animal's location by applying our decoder, namely trying to guess whether the animal is in this location or this location or this location. Which arm of the maze is the animal at, based on the patterns in its brainwaves. And when the animal is running, then what you see is that the waves are really, really strong, and the accuracy of the decoder is really, really good. So there's some sort of connection between high carrier strength and high accuracy. But when the animal is still, what you notice is that the accuracy becomes lower and the carrier becomes weaker. And now I'm plotting the performance of this classifier as a function of carrier strength during stillness. And so what you see is that especially when the animal has a very weak carrier, at those times, the decoder is especially poor at decoding the animal's location. So this led us to try to think about what exactly is going on in the circuit that's preventing us from decoding the signal. And so our initial idea was that there's some sort of global pacemaker that's broadcasting a rhythm that's being picked up by different populations within the hippocampus. So for example, this red population of cells that activates at a particular location of the environment, or a green population or a blue population. And now as the animal goes to the end of the track and this main broadcaster, this main carrier becomes weak, then its rhythm no longer broadcasts effectively to the hippocampus. And so the synchrony that you see within the hippocampus fades. And that might explain why our decoder isn't doing a good job. Now, but there is still the possibility that there's local synchrony within these populations, and that indeed they can support local rhythms, even though there isn't a global carrier around And that's the idea we wanted to test. Maybe even when there is no global carrier, we can recover rhythms that are generated by local, what are called assemblies of neurons. And so if you look at our original pipeline, it relies on this PCA, using PCA to detect a common global rhythm. And we know, though, that when the animal is still, this global rhythm breaks down. But so we don't necessarily want to use this reference as this carrier as a reference. So instead, what we did is derive a technique where we can directly use learn oscillations that aren't connected to a global carrier, but are local, so that you can directly pull out these traveling waves from the raw signal. And that's what we achieved this using a neural network that I developed. It's a complex valued neural network, so it's built to detect waves in patterns of waves within spatially recorded data. And we used this classifier, we trained it on the brain waves, and we tried to predict which arm the animal was in. And then we looked at the local activations of these nodes to see what kinds of oscillations it learned. And what you find is that as you increase the number of nodes in this single layered neural network, you can learn finer and finer grained representations of the animal's environment. To the extent that, so here each color represents the activation of one node, and you find that different nodes activate at different locations within the animal's environment. So this is kind of a proof of concept that this network is able to dissociate all of these different assemblies of the brain into locally active populations of neurons. Now, this is all during running, and what we really care about is during stillness, when we know this global carrier seems to break down. And what we find is that our technique, which is independent of the existence of a global carrier, rescues a lot of the information that our original decoder that relied on a global carrier was missing, especially during periods where the global rhythm seems to be very weak. So to summarize, what I've shown you is that the global rhythm that's identified using PCA can be used as a reference to demodulate and recover information from rhythmic brain signals. But this signal is only reliable when the animal is running. Now, there's another technique which doesn't rely on global rhythms, but rather finds local patterns. And it's able to learn, recover information from these circuits more robustly without reference to this global carrier. And we'll believe that these local rhythms arise from assemblies of place cells. And I think this is really relevant to this crowd because it's one example of how emerging tools are being able to precisely recover the contents of an internal state using measures of collective neural activity. With that, I'd like to thank you for your time. Happy to take any questions, and feel free to email me as well. Thanks. Perfect.
Great, great talk. And two questions for you. One is, I came in as you were discussing Buzaki, and I believe you were citing him for the idea of conservation of rhythmic architecture across species. So first question is, do you generally agree with that, including rats, monkeys, humans? And then two, are you looking at all at harmonic coupling as a key subset of the kind of couplings you're looking for?
Yeah, thanks for these lovely questions. So indeed, the rat is probably has one of the most rhythmic data rhythms that exist. So bats are a notable exception. They have a hippocampus, they have place cells, they fly, and when they fly, their different place cells activate, but they have very weak rhythms. So this suggests that rhythms don't necessarily play, they aren't necessary for the functioning of the hippocampus. Nonetheless, you see bouts of these rhythms across many species. So for example, in humans, you have them during real and virtual navigation. In monkeys, you have them even during eye movements. So as you saccade, you're engaged in some sort of visual travel, and that can engage rhythms. And in a way, part of the motivation of this work is to be able to generalize these techniques so they don't rely on strong rhythms as those found in the rat, and could be extended to creatures such as us that have weaker rhythms. Now, with respect to harmonics, I think that's a really important move forward. So there's labs such as the Wojtek lab that looks at, for example, the fact that the theta rhythm isn't really a sinusoid, but a triangular waveform. And that implies that there's some sort of harmonic in the rhythm. And indeed, there's information in the shape of the harmonics. There's techniques that are now emerging that allow you to learn using neural nets these relationships that extend across different frequencies. The beauty of this network is that when you stay within, you assume one rotational component without harmonics, it really simplifies the learning process. And hopefully with emerging methods, we can extend them to the more general case where harmonics very much exist and probably contribute.
I just wanted to ask, have you looked at how theta rhythms play in like REM sleep in terms of the learning process?
So I haven't. There's definitely a large body of literature where, for example, people recording from the brains of rats while they're engaged in REM sleep, and you certainly see data implying that maybe perhaps if there's anything like dreaming in the rat, if they're dreaming about running through space, but it's kind of, I certainly can't speak to that. I'm not, so can you say more about what you imagine the connection between theta, sleep, and learning to be?
Well, I think maybe it was Gina Poe's lab or something like that, where she was making a connection between theta and learning during REM sleep. And I think the spike was maybe in phase at one point, and then it goes out of phase. And so it's kind of a learning and process is what I was addressing.
Oh, okay. I'm not aware of that research. I'd love to learn more. Maybe you could type it in the chat. Now, one reason I stick to waking states is because we don't have the rat's conscious reports, so all we can do is track them in a physical environment. So I think it would be really interesting to decode these things during dreaming, but it's just a harder problem.
Right, yeah, thank you. I'll send it to your chat.
Thank you.
One more question?
No, not much. Afraid not. Plus, that was a perfect lead into my talk. I'm Jim Pagel. My area is dream science, and I've been studying the electrophysiology of conscious states for over 50 years. In fact, my first paper was the REM sleep and intelligence in mice, which was about, and the way we diagnose REM sleep in mice is with theta wave frequency. So that's been going on for a long time. What I'm going to talk about today is really about dream and how in tying dream into the electrophysiologic brain rhythms. And it's always a little difficult. There's not many of us who are actually dream science. In fact, you could say, is there a dream science? Because almost everyone from every scientific and non-scientific field dreams, and most everyone thinks they understand something about dreams. So I keep my mouth shut at a lot of these meetings as I see people making comments which I think we kind of know aren't true. This And so the electrophysiology of dreaming has kind of, in many ways, gone by the road for many years. Like I said, it's been going on. Our knowledge of electrophysiology and conscious states we've known for an awful long time. And yet the emphasis has been basically on the studies of neuroanatomy. And it's this kind of wonderful situation for me to see everything come back around to the electrophysiology after so much emphasis on the particle basis of the neural network. Ran into this kind of, this is a poster from 1964. And this is a time when electrophysiology coupled with music, coupled with consciousness. And this is a meeting that took place in outside London, includes John Cage, the composer, avant-garde composer, Lamonte Young, the composer and developer of the drone system, where you tune your guitar to the electrical fields of the environment and cut it into segments and double it and play music along that line. They include Bertolt Brecht, and of all people, Alan Watts, the Zen master, entered in. All of this has been kind of lost in space. Now, attempting to integrate the electrical fields with theories of consciousness is something we're doing on a very minimal level, looking at single cell models. The concept of bringing it across with different levels of consciousness is something that's been around, but it's been suppressed in some ways, in that most people have believed that these fields have not been functional. And it's only been in the last few years that there's been beliefs, as we're starting to see, or understanding that electrical fields have functionality. Now, in sleep, we have multiple states of consciousness. Searle, once said that the best way to study consciousness is to study by examples. And in sleep, we have a whole series of examples because of the wonderful situation that we dream throughout sleep. The whole concept of REM sleep and dreaming really set us back in the area of dream sleep and sleep for many, many years, because it's clear that each stage of sleep has a phenomenologically characteristic type of dreaming, and each stage of sleep is defined by a concrete electrophysiologic correlate. So that the forms of wake and sleep consciousness can be phenomenologically grouped based on their association with specific frequencies of the EEG. We'll start off with stage one, or alpha. Stage one is defined, and I practiced sleep medicine for many years, so this is what I did as I looked at these waves tens of thousands of times in patients, that you get a dropout of alpha. So the onset of sleep, you have less than 50% of the page is alpha, but alpha is the frequency associated with sleep onset. Now, sleep onset dreaming, if you'll look in the literature, mostly what you're going to see is hypnagogic hallucinations or parasomnias. But if you wake people up during their first 15 minutes of sleep, you're going to get a dream report in about 70% of cases. In other words, the report of dream recall is as high in sleep onset as it is from REM sleep. Now, I'm going to ask you to kind of think about your sleep onset dreams, because each of you have at least 4 different kinds of dreams, and you can probably call them. If you're sitting, my favorite is sitting in meditation in a very early morning and you drop off. And when you drop off, you will have you will have a quote unquote dream. Sleep onset dreams have the characteristics of they're very intensely visual. There's been studies showing that many of the visual networks of the brain are still active right after you fall asleep. They have very little content, these dreams. They tend to be images. An example would be one where I fall asleep and what I see is an image of the ocean from a different view, where you're seeing the ocean with multiple waves going into the distance, something that you could not actually see that's often from a dream, but it's intense, powerful visual image. And that's a typical sleep onset dream. Other ones of us will have sleep onset dreams driving in the middle of the night on the road when we drop off and you see something that's not in the road. That is a typical sleep onset dream. They can have a huge amount of emotion. The original description of a nightmare from Jones was a sleep onset vision of a succubus sucking his soul out of his mouth. So, but that is your alpha associated sleep onset dream. It can occur, you can have dreams in daydreaming that are similar to such, and you can get alpha intrusion with novel test performance in waking as well. There's also in Labarge, who's, I don't think he's in the room, but you know, he reported that you can get lucid dreaming at sleep onset. About 17% of his reports are from sleep onset dreaming. Now, stage 2, what some people call the junkyard of dream, which is most of the sleeping we do during the night, is light sleep. And the way we diagnose light sleep is with sigma intrusion, 15 cycles a second. And so these waveforms, you'll see these bursts of sigma is what stage 2 is in light sleep. Now, stage 2 dreams are quite different than what we're talking about stage 1 dreams. Light sleep dreams tend to be very day reflective. They don't have a lot of import or interest. They're not life transformative dreams in any form. My favorite stage 2 dream is one I had one time where I remembered a number, or I had a number in a dream, and that doesn't happen to me very often. And it really crossed my mind that I should really remember this number, because, you know, it could be like a lottery number, you know. So, but I didn't have the energy to get up and write it down. And I spent the entire night wandering in and out of this dream. I remembered it again. I remembered it again. I remembered it all through the night. And of course, I woke up in the morning, it was my phone number. But stage 2 dreams tend to be that kind of reflective kind of pattern, though they can be intensely emotional as well. Individuals with panic attacks will have panic attacks in stage 2, those brief moments of terror. It's also, we know that stage 2 or light sleep is associated with memory consolidation, perhaps more than any other stage of sleep. Now, REMS is one of those things. REMS has been kind of a problem for many years, and it may be an artificial construct. That's after 50 years of looking at it. I'm not sure that REM sleep is different from non-REM sleep, except that it's a different frequency. And part of the problem has been that when we do EEG studies in humans, we attach the leads to the scalp. And when we do EEG studies in animals, we implant them in the brain, okay? And they look totally different. So you don't see theta frequency in humans. This is a typical pattern. You get eye movements. The EEG looks like a wake. You don't get frequency patterns in the human. For some reason, and this is unclear, and I've never heard a description of why, theta doesn't conduct across the scalp. It doesn't go this far in the brain. It's something you see in animals because you're using inserted electrodes into the brain to record animals, because you don't put little scalp electrodes on because they shake them off. So if we would stick electrodes in humans, we would get a different report. I'll show you. This is kind of great fun to be able to put up a slide from a study that I did 50 years ago. So this is the REM sleep and intelligence in mice. And basically what we found was that mice breeds who were better at running mazes had a higher percentage of REM sleep than mice breeds who perform more poorly at running mazes. So that was that's kind of the original paper for studying learning and memory and associating with REM sleep. But the association is basically with theta wave frequency, because when you put these electrodes, we'd see these beautiful runs of theta occurring to diagnose REM sleep in the mouse. We don't watch the eye movements. Some people will put an EMG on, like in this one, but basically what you're seeing is a huge spike of coherent theta wave whenever REM sleep is occurring when you study the mouse. Now, in humans, if you put in interthecal electrodes, and there are studies that have been done now, mainly in doing studies of individuals who are going to have seizure surgery, where they leave electrodes in place and we get to watch sleep with an electrodes down into the brain. And what we find is a theta frequency. So humans are just like animals. We have theta frequency associated with our REM sleep, but we don't pick it up on the surface of a scalp EEG. If you look on the power frequency, you're seeing frequency occurring at theta way down here on the left side. This is a gamma frequency as well that occurs associated with REM sleep in the human and in animals, that you don't see any of that when you look on the waking EEG. And if you look at both beta and theta frequency, when you put an interthecal electrode into the human brains, you're going to pick up theta wave frequency in 80% of your inserted electrodes. So one of the things about, so in REM sleep, basically is not a paradoxical state. It is not a state that is non-synchronous. It is a synchronous state of theta rhythm, just like sleep onset is a synchronous state of alpha rhythm, and stage 2 light sleep is a synchronous state of sigma rhythm. The type of dreaming that you get in REM sleep, the major characteristics that differentiate REM sleep dreaming from the dreaming of the other states is length of report, and the other one is the association with nightmares. Everything else, content, bizarreness, none of those hold out as being, at least when you take out transference problems, you don't get a difference in dreaming between the different states. So you get a high recall, you get 70% just like at sleep onset, get a longer report length, and you get intense emotions. A typical REM's dream, I'll give you one of my great nightmares from when I lived in Hawaii. I'm walking across a taro field with my wife and daughter as the sun sets in the distance through the water and the mud between the plants, and I'm walking along, and lo and behold, here comes an incredibly large snake, and there's nowhere to go and nowhere to turn, and not only am I going to be attacked, but the people that I'm protecting and depend on me, I'm going to lose them. And but you know, I spent my career working with PTSD primarily in patients. And so I teach people to do lucid dream conversion. So what do I do? In my dream, I have a lucid spell, and I turn the snake into a blow-up animal, you know, from the pool, and suddenly everything's wonderful. Of course, I'm not a big great theorist. I think dreams are really important, and I think they have information. And if you lucidly change your nightmare, you may lose some really good information about what's happening in your head. I did that with that dream, but that is a nightmare, and that was REM sleep. And except in patients with PTSD, REMS is associated with nightmare, is associated with theta, and is associated with gamma frequency. In meditation, we get alterations in the theta alpha ratio when we do the QEEG analysis. So we can alter these frequencies as well, just like we can alter them in lucid dreaming. Beta, we get with focus waking. Everybody's talking about beta and gamma as a frequency. I wanted to include one more comment about beta and gamma because we don't see it on human EEGs. And it's for another reason, not just the interthecal insertion, not that we don't stick electrodes in the brain. We don't see it because we use 60 notch filters on all our systems. The problem is that even when we're declaring someone dead in the ICU and we hook up an EEG, we put on a machine that clears out and blocks all the 60 cycle because the environment is just full of electrical systems going off, and it just overwhelms the system. So almost all human EEG analysis is done with a 60 cycle block in place, and that very often takes out gamma. So you don't see 40, 50, 60 cycle because you've got a block in your system. And so it's wonderful. You can see it in animals, and you can see it in interthecally placed electrodes in humans, but otherwise you're not going to see gamma frequency on a regular occurrence. Delta is my favorite state. There, you know, for years there were people going around saying the way you diagnose deep sleep is that, in fact, I heard Christoph say this at his last presentation a few years back. Deep sleep, no dreaming. That is so much bull. The situation is that individuals who have clear deep sleep parasomnias have dream reports at least half the time. So if we have someone who is sleepwalking and you wake them up, they will have a report of the dream. If you have someone who has a night terror, half the time they will have a report of a dream. It's often very different than a REM sleep dream, or a sleep onset dream, or a light sleep dream. What it is an awareness of dreaming, usually intense emotion, color. I'll give an example of, once again, because most of you have had a deep sleep dream. The characteristics of a deep sleep dream is it's more common. The parasomnia is, deep sleep parasomnia is confusional arousal. Night terrors and sleepwalking are much more common in childhood. So these are very often childhood dreams. They're often repetitive. The one I had as a child is that I'm in a chamber underground, and there were 3 giant stones that are rolling synchronously round and around and around in a chamber that's like green, covered with moss, with the water dripping, and they start to totter. They start to go out of oscillation. So one starts like this, and all of a sudden they all just deconstruct and fall apart, and everything blows up, and I wake screaming. This was my night terror. This is a classic night terror. Now, many of you will have had similar experience. I've worked with artists in their dreams, and very often artists will incorporate early experiences of night terrors into their art. It's something that sticks with you your entire life. But the formal characteristics of a delta dream: fragmentary recall, extreme emotion, fear, disorientation, confusion on arousal, even autonomic behavior. Now, the latest area that's been developing, and this is more into the waking area, though Domhoff has been really pushing the association with subdelta frequencies and dreaming as well. But we didn't even know about subdelta frequencies. It's not something we see on a waking EEG. You have to do a quantitative EEG analysis. It wasn't even picked out until late in the 80s. And now this is how we diagnose the default network. And so we're in a situation now where the operation of waking behavior that is default, mind-wandering type of behavior associating widely separated areas of the brain seem to relate to a subdelta frequency occurring in the brain. So basically, this is my conclusion. The EEG frequency electrical fields are basic diagnostic, stage-specific attributes of the different forms of sleep consciousness. REMS is neither non-synchronous nor paradoxical. It is a synchronous state characterized by theta and beta frequencies, and the synchronous CNS electromagnetic fields are a primary attribute of consciousness. Thank you. So it's Dr. Gennaro. You're up next.
Gennaro, yeah.
Dr. Gennaro.
Gennaro, yeah. Can you hear me? Can you hear me?
Go ahead, and you'll need to self-introduce because I don't have.
Right. Let me share the screen first.
Okay.
And I'll keep my picture up there. So let's see, slideshow. So my name is Rocco Gennaro. I'm at Southern Indiana, University of Southern Indiana, in Evansville, Indiana. My talk is called Consciousness and the Prefrontal Cortex. Now, I am a philosopher, not a scientist in that sense, but obviously I'm in this session for a reason. Part of the reason for the philosophical aspects of it is there is a theory of consciousness, philosophical theory, which comes up in this issue. So I just have a slide here for the basic summary, but it's really just the abstract. There is a continuing debate about to what extent, at least, the prefrontal cortex is necessary for having conscious mental states. So I look at some of the evidence, brain imaging, anesthesia, lesion studies, and so on. And then I also, like I said, examine these findings with an eye toward how the data might bear on what's called the higher order thought theory of consciousness, which I previously defended. I mean, I don't think that the prefrontal cortex is required for having conscious states, but the reasons are also interesting. Sometimes the idea that conscious states require some prefrontal cortical activity is thought to cause trouble for the theory, and I'll explain as we get to it. Terminology-wise, you're probably mostly familiar with that Nagel's famous phrase, what is it like? Consciousness. I'm talking about subjective experience, right? And we also sometimes want to distinguish between when we talk about a mental state being conscious, as opposed to sometimes we'll refer to a person or a creature or an animal's conscious, so that kind of thing, creature consciousness. So just to get a little background on the theory that will come up, philosophers, of course, are interested in what makes a mental state a conscious mental state. How do we distinguish between unconscious mental states and conscious mental states? One way of approaching this issue has been through what's sometimes called the transitivity principle. conscious states or mental states that I am aware of in some sense. I mean, it almost at least sounds kind of common sense if you think of it that way. On the other hand, an unconscious state would be those mental states of which I'm not aware. So now I'm not going to go through all the reasons, but for a lot of reasons, some philosophers think that this higher order awareness, if you want to call it that, is best understood as a thought composed of concepts. And so hence the name higher order thought theory in this case, right? So the higher order thought theory says that when you have a mental state that's conscious, there's a suitable higher order thought directed at the mental state, which of course, if you're thinking about in terms of areas of the brain, it looks like it certainly implies that some more advanced areas of the brain might be implicated, right? And higher order thoughts really are just what a lot of times in psychology referred to as metacognitive or metapsychological state, mental states directed at other mental states. If you like a figure, what I've described is pretty much what's on the left. So, if I'm having an outer directed visual perception, I have the world directed conscious state, and there's a hot, unconscious hot. However, sometimes, we turn our attention inward, right? In what we sometimes might call introspection or reflection. In that case, what you find here is that the unconscious hot itself becomes conscious. And so, at least according to the theory, you then have your conscious higher order thought directed at the mental state, not at anything outside, right? So if I am consciously aware of a desire of some kind, then I have what might be termed an introspective state. So this is really, the next slide is really what I just sort of mentioned. There are reasons for it, a lot of obvious reasons, right? I mean, you don't want there to be circularity and infinite regress and all of that, but there are other reasons for it. So it's at least good to keep distinct first order conscious states from when cases where the higher order thought itself is conscious in introspection, right? Now, most at this session, of course, will be very familiar with something like this, but these are some of the areas of the brain that I might refer to later. The most obvious one, the prefrontal area, right? And some of the other areas that are brought up a lot, visual cortex, and so on. A little more specific, frontal cortex. Of course, there's many regions, subregions of the prefrontal cortex. And there's also the ACC here, anterior cingulate cortex, which, I don't know, depending on who I read, some seem to treat that as part of the prefrontal cortex, some not. I tend to think it's not, but I'm not sure I can give a good reason, but I've read some that don't include it as part of the prefrontal cortex, and others who say, well, it's kind of is, right? So in any case, there are still multiple other prefrontal regions, and there are other areas, of course, further back in the brain and deeper in the brain that do come up as well. So we'll look at anesthesia is one place to look. Obviously, it's kind of important to be able to tell from a third-person scientific approach whether consciousness is correlated with specific brain activity. This is, I tell my students, this is not just some kind of esoteric exercise. If you're an anesthesiologist, you want to be able to accurately determine whether a patient's having conscious states, such as pains, right? If you make a mistake there, it can have catastrophic results. And it seems from the research I've read and things I've seen that the effects of some kinds of anesthesia kind of lend support to the notion that it's really neither the basic sensory areas nor the prefrontal cortex area that seems sufficient to produce conscious states. Now, it's not that simple, of course. There's always complexities, but if you take a look at some of the evidence that I've put forward from various papers, Schroef paper, results show that deep sedation due to propofol, which is an anesthetic associated with reduced interactions between the associative cortices. However, the functional interaction of parietal areas were deteriorated to a significantly larger extent than the frontal, which is the area that I'm most interested in here, but also temporal areas. Alkire, Hudetz, and Tononi unconsciousness is likely to ensue when a complex of brain regions in the posterior parietal area is inactivated. So it looks like when that is suppressed, that is where consciousness seems to kind of go away, right? The evidence, another quote from Hudetz, the evidence overall shows that anesthesia primarily causes the suppression of feedback and top-down brain mechanisms. In other words, it is something further up in the feedback, if you like, or the feed forward, as opposed to feedback loops. Networks based on the posterior parietal cingulate precuneus region as a hub, the nonspecific thalamus are putative candidates for the neural correlate of the state of consciousness. Notice again, there's no mention there of prefrontal cortex. More recent paper, Hutt and Hudetz, they demonstrate, talk about a couple of studies, one of which demonstrates in surgical patients that anesthetic invariant electroencephalographic effects occur in cortical top-down connectivity. And here, talking about ketamine, not propofol, but is found to suppress frontoparietal functional and directional connectivity similar to those produced by propofol. Now, sometimes a fronto is vague. It's just prefrontal or just generally front areas of the brain. Sometimes they mean something very specific. Hudetz and Mashur, who are they're both there, I would imagine. Investigations to date converge on the conclusion that with various anesthetic agents, a depression or disconnection of lateral frontoparietal networks appears to be a consistent correlate of disrupted connected consciousness. After a dose of propofol, highly connected hubs in the brain undergo a reconfiguration with connectivity patterns in the posterior parietal cortex being disrupted. Here's at least one of many interesting results. If you look at, say, A and C, left to right here, you see that the wakefulness state of the visual cortical area, sensory area, there's two different views, right? Top side, right? You look over to the right and you see, even in cases where anesthesia has been applied, there really is anesthetic-induced unconsciousness does not seem correlated with the inactivation of the primary sensory cortex. So it does seem at least reasonable to think that it doesn't suppress, the anesthetics don't suppress necessarily the basic sensory areas. And then there are other reasons to think it doesn't suppress the prefrontal cortex either. The bottom pair, B and D, are the auditory. It's like the auditory parallels. So if you see, you see the relative preservation across states in those primary, in the case of A and C, primary visual cortex, primary auditory cortex when anesthesia is applied. So again, as far as the theory goes, hot theory, it looks like anesthesia doesn't mainly target prefrontal areas. We have other reasons for this coming up. But it might make sense, of course, for a higher order thought theorist to look elsewhere in the brain for where many of the higher order thoughts might occur. So I mean, I've argued that in past writings. Some other philosophers have argued, higher order theory implies that the prefrontal cortex must be involved, and that makes it less plausible. It doesn't seem like, or something of that kind. And I argue that higher order thought theory doesn't need to require that sort of thing. Right? Still, it is likely true that the prefrontal cortex is required for having more sophisticated introspective states, but this is not a problem for the theory because, again, just to have a basic first order conscious state does not require the more sophisticated introspective state. So that comes up as well. No doubt prefrontal cortex about more advanced reasoning, reflection, and so on. but that's not required for just basic outer perception. Certainly not required for something as basic as pain and sensations and so on. There are also independent reasons to think conscious states can occur in the absence of prefrontal cortical activity, right? So even going back a little ways, there's some evidence that conscious experience isn't eliminated entirely, even much when there's extensive prefrontal cortical damage. Matter of fact, in some cases, large areas of the prefrontal cortex can be removed with little change in at least some conscious perceptual experience, right? Even when you think about the damage of going back to the famous Phineas Gage kind of case, or lobotomies, right? Things of that type. Also, when subjects are highly engaged in a perceptual task or absorbed in watching a movie, there's widespread neural activation, but little PFC activity. So, Rafi Malik and others have done some research along these lines, where, in other words, when the person is especially kind of instructed to focus outward, not reason as much or not think too much, you don't find too much prefrontal cortical activity when they're absorbed in a task of a certain sort. Now, there are some other studies that do seem to implicate prefrontal activation, but the usual response, or kind of response is given, is that arguably this occurs because of the need to subjects to report their experiences. And the prefrontal cortex is likely to be activated when there is reflection or introspection. In other words, if you know that you're going to be asked questions about something, or give a report of some kind, or even answer yes or no questions afterward, you might start using your reasoning, you might say, as you are going through some kind of task or doing a certain kind of thing. And if you look closely at some of those studies, Dehan and Chanzhou, they themselves actually concede that their experiences, that their experiments, do not suffice to establish that there's a frontal contribution is causally necessary for conscious perception. Many times in their studies, they actually identify parietal cortices instead of the prefrontal cortex, right? And you find something similar in other cases. The main thing to keep in mind, though, is that at least in terms of the philosophical theory is in the background here, if you are performing a task of any kind, and maybe you know that you're going to have a task, something to answer or introspection afterward, that doesn't really cause a problem for the theory. The theory does not say conscious states require introspection, just a higher order thought of some kind, right? Nguyen and Vogeli cite numerous experiments having to do with different degrees of self-consciousness, ranging from some very minimal to some very sophisticated. I won't go through all of everything on this slide, but there are some studies that suggest, again, that, for example, the parietal cortices, temporal parietal, again, posterior cingulate cortex, and then down here, as I mentioned earlier, the ACC seem implicated much more than the prefrontal cortex. You have some also very recent work, Rebecca Sachs and others, mind reading, theory of mind area, so to speak, in the brain. Seems kind of sophisticated, like a kind of self-consciousness or thought about thinking, right? That sort of ability, that this kind of meta representation is located in the anterior cingulate cortex, or one prominent area mentioned a lot is the temporoparietal junction, right? TPJ. So lesion studies is another thing that is looked at for these purposes. Benji Kozic has done a lot of work in this area, and he's also pointed out, but I have too, that sometimes when people think of themselves as prefrontal theories of consciousness, they don't really mean that they're necessary or constitutive of consciousness. Sometimes when we look more closely at the results, they use phrases like PFC lesions can affect conscious experiences. And that's, of course, a much weaker claim, right? And the same kind of thing is said in other places, right? So once again, it seems even major lesions in the prefrontal cortex typically show no apparent degradation in consciousness. That's a more recent paper, 2021 paper. In many visual masking and stimulus detection tasks, PFC lesions do not result in deficits in identifying shape, color, or location of stimuli. Now, Michael and Morales respond to this so-called argument from report. You know, the idea that, well, it's probably when the PFC activity does show up, it's really mainly due to this notion that, well, you know you're going to be reporting on it or ask questions or that kind of thing. And they don't think that argument is enough or is a good counter to it. So they reply to that, you know, counter reply, if you like. They worry that some of the experiments they're talking about don't require reports. Maybe there's a worry about a null hypothesis, you know, false negatives and so on. They also mentioned in many trials, subjects are asked to report about both the conscious and the unconscious. Like, do you see anything? And sometimes the answer is, you know, is no, right? Something along those lines. How can you explain this? And working memory, it is pretty well understood that is oftentimes at work for both conscious and unconscious information, right? Now, you know, in response to that, sometimes it's said, and this is some things that have occurred to me too, but surely introspection and reasoning can be a confound even when there's a no report or detection task is very basic. Some reasoning might still occur even if no reporting is expected. Just because, in other words, a task doesn't require reporting. Well, thank you very much. We would surely almost always do it to a certain extent. The reporting decision also need not is sometimes is if it's a simple yes or no detection, they occur at different times. So that doesn't always correlate with the seen versus unseen timing. It doesn't seem like it does. When you see the stimulus, your response is immediate. It can be a yes, but when a no is not usually given until the end of the trial, because you have to kind of keep waiting, right, to see if you're going to see something. So the difference in reporting may not be due to a conscious vision as such, as much as anything, expectation or something like that. And then very recently, there's this study about intracranial electrical stimulation. And I'll just throw this up there and get what we can out of it just as I conclude here. I mean, again, in a lot of cases, say the blue here, where it's clearly prefrontal cortex, you know, not really many known effects in terms of this procedure or this method, the IES method. Same kind of thing, you know, down here, the V ventral. Sure, there are other areas, but not many in the prefrontal cortex. Perhaps some conceptual thought. Perhaps maybe the strongest case is some emotional phenomenon appear when you have this stimulation in the prefrontal cortex, but it's not as much as you might think. Most of the others are, you see, sort of inside past the prefrontal cortex. And so there are always further questions, but I don't want to go over time. So some of them I mentioned already. And, you know, conclusion is really sort of a repeat of the abstract. It just seems that the evidence seems to show that, to me at least, prefrontal cortex is not required for basic sensory conscious states, or that hot theory is should be committed to the view that it is, contrary to the claims of both critics and advocates of hot theory. I'll just leave it at that. Thank you. Just a couple of my recent books, and I'll just leave it at that. Thanks.
Hello, everybody. My name is Jim Beran. I'm an independent researcher who lives in Monterey, California. And you can see a glimpse of that in the slide. Here's a quick preview of what we'll talk about. Our general topic is hypotheses for biological evolution of consciousness. and we hope to find better hypotheses. 2 years ago, at the virtual Tucson 2020, we proposed multiscale hypotheses with 4 scales. Those hypotheses did not explicitly mention quantum effects, but this time we're proposing 5-scale hypotheses. First, one that doesn't include quantum effects, and then one that explicitly includes quantum effects. We'll show one way of adding scale-specific quantum effects to a multiscale hypothesis. And we'll discuss the result. In case you're dying of curiosity, I'll show it to you now, and then I'll tell a story about how it came to be. There it is, our destination, a 5-scale hypothesis for evolution of consciousness, including explicit scale-specific quantum effects. At first glance, it might look intimidating, but we'll go through a story of how it came to be. After that, I hope it will seem more approachable. And my thanks to those listed here for their help. And also special thanks to 2 others. My longtime collaborator, Joni d'Arc, an above-average Norwegian rat of French descent. And what makes her above average? Not only is she a master chef by day, but at night she works as a neuroscientist studying the biological evolution of consciousness. And thanks also to our new collaborator, SCAT. When SCAT showed up looking for a job, Joni hired him to be maitre d'. Like Joni, SCAT has other interests. He's fascinated by quantum mechanics, especially by quantum effects. When he heard Joni discussing her new 5-scale evolutionary hypothesis, he made a paw-waving argument for adding quantum effects. Some background: researchers have proposed that various genes and proteins affected evolution of consciousness. Joni's previous flash of insight was that these proposals are not all inconsistent. Multiple genes and types of proteins could play roles in evolution of consciousness. Also, Joni couldn't accept that evolution simply stirs up genes and proteins as if it were making ratatouille. Ratatouille isn't even on her menu. Instead, Joni's anti-ratatouille conjecture, or ARC, proposes that the evolutionary path included gene-protein change so that proteins interact in new ways. This table shows features of a multiscale hypothesis with a top row for scale, a middle row for type of change at each scale, and a lower row for possible effect from each change. Let's use the table to summarize features of a 5-scale hypothesis for evolution of consciousness. At the smallest scale in the table, genome variation, mutation in germline DNA affects gene expression. Since genes are expressed as counterpart proteins, a possible effect is a changed protein. Next, at subneuron scale, changed proteins interact in new or changed ways. A possible effect is a new feature in cells in which the interactions occur. Next, at neuron scale, the new or changed protein interaction relates to output patterns of neuron action potentials. A possible effect is specialization of key neurons to provide output patterns that control other neurons. Next, at superneuron scale, the changed output patterns from key neurons relate to change in operations of neuron groups they control. A possible effect is that groups of neurons operate in a coordinated manner, such as by being synchronized. And finally, at brain scale, the changed operations of groups of neurons relate to change in brain activity. A change in brain activity might correspond to an evolutionary change in an organism's conscious experience. Now, as an aside, in the second row of the table, note the upside down V symbols just above each proposed type of change. In logic, the upside down V, sometimes called wedge, is the symbol for the conjunction or and operation on propositions. In this table, the wedge symbol means that all 5 of the proposed changes or sub-hypotheses in the second row must be true for the overall hypothesis to be true. We call this type of hypothesis a conjunctive multiscale hypothesis, and we can use the summary in the table as a form or a template for a detailed conjunctive multiscale hypothesis. But why would one want a detailed conjunctive multiscale hypothesis anyway? Well, since 2020, Shea Joni's menu has included hypotheses about evolution of consciousness with change in protein interactions. And these hypotheses range in detail from less to more detailed. As shown here, detailed multiscale hypotheses with protein interactions can be bolder and more falsifiable, and they can be better if they more closely follow the actual evolutionary path. Now, Joni has added a new 5-scale hypothesis to her menu with features like those we just discussed. Joni's new hypothesis is tied to the axon initial segment, a region of the neuron shown illustratively in the small diagram at the top of this image, and often abbreviated AIS. The AIS is part of the axon that initiates a neuron's output action potentials. And here we see some of the relevant proteins and protein interactions in a neuron's AIS. Here are brief descriptions of 3 categories of proteins and their interactions. A protein named tubulin, which you've heard a lot about already, forms microtubules, shown in gray at the bottom of this image. The microtubules are central to the structure and operation of eukaryotic cells. Anchorin G molecules, shown in red, extend from the microtubules to the membrane around the AIS. And connected to anchorin, but extending through the membrane, a light blue voltage-gated sodium channel labeled NAV is an example of an ion channel. When concentrated in the AIS by anchorin, ion channels can initiate action potentials. Going to a different set of features, this image illustrates polyglutamylation. Try saying that quickly 5 times. Like other proteins, tubulin is produced from DNA, a process that includes translation to a chain of amino acids. After translation, the resulting tubulin can be modified by attaching or removing amino acids or other components, often in the form of side chains that extend outward from the main chain. These modifications are called post-translational modifications, abbreviated PTMs. And glutamylation is a PTM that adds an amino acid named glutamate to tubulin. If more than one glutamate is added, that is called polyglutamylation. Polyglutamylation is abundant in brain tissue, and it adds negatively charged glutamate side chains on microtubules. We call proteins that regulate post-translational modifications PTM-reg proteins for short. And this diagram shows three types: CSAP, TTLLs, and CCPs. They are believed to regulate the amount of glutamylation as shown in the diagram. Three levels are shown: low, intermediate, and high, with polyglutamylation being especially frequent at the high level. The green arrow in this diagram points out the high level of polyglutamylation at right. You'll see the chains, the side chains, have several glutamates. So Joni's 5-scale hypothesis proposes that polyglutamate tails on microtubules provide electrical and/or magnetic interactions between proteins. Now you've had a quick introduction to the AIS. Here's a quick summary of Joni's new 5-scale hypothesis. Note that the hypothesis includes a change at each scale from genome variation scale up to brain scale. And the changes are all related, from mutations through proteins and other polypeptides such as polyglutamate tails, to patterns of action potentials from key neurons, to changed operation of neuron groups, and finally to change in a brain's electrical and/or magnetic pattern, tying into previous talks about the EEG. This change at brain scale corresponds to an evolutionary change in conscious experience. Let's listen in on a conversation that occurred when SCAT learned about Joni's new hypothesis. SCAT didn't like the new hypothesis because he couldn't find any quantum effects in it. Well, Joni argued that there were implicit quantum effects. Quantum effects are always occurring, for example, among subatomic particles. But SCAT was insistent. He wanted explicit quantum effects in the hypothesis, and Joni was stumped. How could her hypothesis include explicit quantum effects? SCAT immediately had a bright idea: multiscale quantum effects. In other words, the hypothesis could include scale-specific quantum effects at more than one of its scales. But why include scale-specific quantum effects in a hypothesis? And how would one do so? SCAT quickly came up with answers. In response to why, his first few reasons for adding scale-specific quantum effects might look familiar. They're similar to the advantages we discussed earlier for a detailed multiscale hypothesis, including more falsifiable, bolder, better. But SCAT's reasoning also took an unexpected paw-waving leap. If conscious experience is difficult or maybe impossible to explain as a classical effect, then he proposed it must be a quantum effect. In answer to the how question, SCAT proposed a three-step approach. First, add a quantum effect change at subneuron scale, specifically a change in protein or polypeptide folding. Second, add a quantum effect change at brain scale, a change that corresponds to an evolutionary change in conscious experience. And third, add suitable quantum effect changes at intermediate scales, such as at neuron and superneuron scales. To help explain SCAT's approach, here's that table again, but with a new third row to show changes in scale-specific quantum effects. The first addition to the third row is a change in polypeptide folding at subneuron scale. The folding could, for example, be in a protein such as an ion channel or in a short polypeptide such as a polyglutamate tail on a microtubule, or in both. Second, SCAT adds a change in a quantum effect at brain scale and specifies that the change in the quantum effect corresponds to an evolutionary change in conscious experience. Note that SCAT has conveniently ignored that the brain is warm and wet. He's just assuming, for the purpose of argument, that there are quantum effects at brain scale. And third, SCAT adds changes in quantum effects at the intermediate neuron and superneuron scales. At these scales, SCAT hopes to add quantum effects that approximate and are consistent with a continuum of quantum effects across a range from subneuron scale to brain scale. SCAT takes inspiration from a suggestion by Bowman-Hiley in The Undivided Universe at page 160, where they write, "There's a continuous transition between this total quantum reality and the large-scale world of ordinary experience." Note that the second row of the table still shows 5 changes from mutation on up to brain activity, and the third row now adds 4 more changes for a total of 9. And also that all 9 of these changes are logically ANDed to form a conjunctive multiscale hypothesis with 9 subhypotheses. This slide simply illustrates that falsifying any part or subhypothesis of a conjunctive multiscale hypothesis falsifies the whole thing. You can falsify it by disproving any of the classical or quantum effects it proposes. So here we are again. Remember this is SCAT's version of Joni's 5-scale hypothesis, the destination we briefly discussed at the beginning. Now you know where it came from. Just a few quick points. The added quantum effects in this hypothesis are paired with classical effects from Joni's hypothesis. Each quantum effect can be seen as correcting or fine-tuning its paired classical effect in a specified way. And we suggest that each quantum effect might be a non-local correlation in its respective scale. At brain scale, the quantum effect change occurs in a Durbridge C field. This hypothetical quantum field results from suggestions by my friend Dr. Tim Durbridge. He suggested that we need a new physics to explain consciousness, and this is my attempt to provide one without filling in any of the details I might point out. Events in the Durbridge C field can correspond to features of conscious experience, such as transitions from one experience to another, for which I use the term dynamic conscious experience because it has those transitions. Here we have a brief comparison of Joni's and SCAT's hypotheses. Reasonable minds could differ, but I think SCAT's is better based on falsifiability, boldness, and maybe explanatory power. Here are our inclusions or conclusions. And here are some thoughts about possible future work. Thank you for your attention, and please contact me by email for access to a video file of this talk or my 2020 talk, or for complete citations. Do we have time? Oh, thanks. Time for questions?
I'm going to talk about a set of experiments that are intended to test a theoretical prediction out of a framework that came out from a model of spatial and temporal summation. So the idea here is, so our lab, so Erik and I collaborate at UC San Diego. I'm in bioengineering and neurosciences. Erik is in neurosciences, and so my lab basically uses math and engineering to study the brain as an engineered system effectively. And what I'm really interested in is the algorithms that the brain uses to compute. Can we abstract away as many of the biological details that are important for implementing those algorithms in the wet, squishy wetware of the brain? But can we abstract those away and keep the mathematical rules that underlie those? So we have a particular interest in the role that the geometry of biological neural networks plays in constraining and defining dynamic function. So what you see there, just by way of example, is this is a simulated network of 100 neurons, and they're all the two plots on the right there are two raster plots. The only difference between, and I assume everybody here knows what a raster plot is, there's time on the x-axis, neurons from 0 to 100 on the y-axis, and every time you see a little dash mark, it means that neuron fired. The only difference between these two raster plots is we changed the signaling speed at which information was propagating in this network. And so it turns out that for the same geometry, if you mess around with the signaling speed, you're creating a mismatch between the rate at which information is being processed locally in an individual neuron relative to the rate at which information is propagating across the network. There has to be a balance in that. There has to be a balance in that ratio between local information processing and global information processing on the network. And so we've shown theoretically, we've actually proven mathematically that the efficiency of a network will break down if this principle is violated. So if you look at the top there, a real neuron, you can see there a morphologically reconstructed neuron. If you think of that as a little graph, as a network, it doesn't have straight line edges connecting those vertices, those nodes. And there's a reason for that. It's actually, it's modulating the delays in that cell to preserve this ratio. And so we've shown this with, in this one paper in Nature Scientific Reports, we actually showed that a neuron seems to optimize its morphology, its shape, to preserve this refraction ratio. And where we have a hypothesis that, in particular, for example, in neurodevelopmental disorders, independent of the biological, for example, autism is typically studied by, cataloging genetic changes that occur. The hypothesis here is that independent of the genes that are being changed, what's actually being affected systematically from an engineering perspective is precisely that it's altering this refraction ratio, this balance between local energy processing, information processing, and the speed at which is happening at the global scale in the network. And so we're starting to look at this in human-derived brain organoids, for example, derived from autistic patients. And most recently, we started looking at it in humans. We've carried the same type of analysis out in the C. elegans connectome, for example. C. elegans is this famous worm, right? A very famous model of neuroscience. And we were able to show that the wiring diagram of the worm is actually seems to be have evolved to support certain types of function, again preserving this idea of a refraction ratio. And most recently, we've now starting to do human studies using MEG to test this ratio. And Erik's going to talk about the protocol we're developing to test this in humans.
Thanks, Gabe. So I'd just like to make a little comment that it's a real privilege to be back at the Science of Consciousness. And I hope Stuart gets the chance to hear some of our talks. and for me to say thank you to Stuart for putting this together. It's really amazing to be with people again. It's kind of funny that this is the second in-person real talking meeting that Gabe and I have had in the last 2 years. And so coming to a conference and being able to give your ideas, but also to be able to talk in the halls is a real privilege, and it's something that I'm amazed that I've missed. So Gabe has talked about how he discovered this speed limit. How do you move information across a given distance and match the speed? And oh, there's Stuart right there. And Betsy. Stuart, thank you so much for doing this for us. It's a privilege to be here again. And I just literally was just giving thanks for you being here. It's awesome to have you. So controlling speed over distance is this discovery that Gabe has made, and it may well be help us solve this classical problem in consciousness and the human brain called the binding problem, where we link information together across the vast areas of human brain. And so all kinds of different things need to be linked. And we talked about this earlier. We've heard about emotions and thinking and consciousness. And the classic experiment by Libet told us that timing was important, that when you touch your fingers together, the time that it takes for your information to move through your body and through your mind and come up with that integrated conscious experience is timing controlled. And so where is this timing control? And Gabe has just put it together that in the C. elegans, he has seen that the wiring of the neural connections are matching time over distance. So everything all arrives at the same time, despite these sort of chaotic looking neurons, these funny looking things that, in contrast to a computer chip, which have nice straight lines and rows and columns and link up all nice and synchronously. So to me, one of the greatest conundrums is visual conscious experience. We have this experience all day, every day, through our lives of this integrated visual experience. And we have the classical description of how the brain does this. So we have light coming in and it hits the back of the retina through focusing, and then it's passed through these crazy pathways through the center of the brain, through the thalamus, reaching the primary visual cortex, and then propagated out further into the brain. So again, just not looking like a highway at all. How can this system put all these different bits of pieces together and give us our integrated conscious experience? That's the question for me that that we're working to address at the human scale. So instead of being able to look at microscopic assemblies in a petri dish or in small organisms, we want to be bold and look across the human brain. So remember, at the back of the head in the visual cortex, primary visual information is put together. And we're looking at things like looking for edges. So we see in the central diagram, the little blue box, the edges of the chin on the statue there. That's what the pieces of the visual cortex are looking at. And it's this model of the red cells linked together, and then an edge comes and they fire the cell in the V1. So how are these bits and pieces all linked across the brain? And I in preparing this talk, I found this fixed section, and I apologize to my wife who's in the audience, who thinks these things are kind of gross, but they are a marvel. And what in the blue, we see the line of Gennaro, and the visual cortex is called the striate cortex. And you can see, in contrast to all the other gray areas that are abutting to the white, the gray and white matter, the striate cortex has a stripe. That's why it's called the striate cortex. So this is the where the visual outputs from the eye through the thalamus come and reach the cortex of the brain in this striate cortex. And look at the bizarre shape of that through the gyri and sulci. But then there's the most important point. It's divided immediately in half. So as we know from lesions that occur in people, if they lose one half of their brain, they still see the other half. They see the other visual field. But our brains knit this together all the time. We have this constant knitting together across the midline. So things go to, signals from the eyes go to the left and to the right. And yet we don't have that percept. There is no line. There's no line in the middle, like, you know, when they put in a sports stadium, when they put all those things together and the screens together and you see the lines, there are no lines. How is this knitting together? And my suspicion is, and our hypothesis is, that the refraction ratio is part of what is done. So in other words, there's a strong organization of the signals propagation, and the place we want to look is across the midline versus in an immediately adjacent area. So it's a little bit dangerous to come to a conference and talk about what you're planning to do. And our dream had been that we would be able to have some data to talk about at our conference here. We are doing recordings right now. So I'll show you that this is a MRI image. And what we're going to do is see these yellow highlighted areas. That's this striate cortex. These are the visual areas of the brain. And so this is coming from the back of the head. And then if we look through the plane demarcated by the red line, we see this side. You can see the visual cortex again here. So we're moving towards the midline. So all this is striate cortex that's being identified. So we have highly nuanced ability, high resolution ability to detect the specific anatomy that we're looking for. And then the exciting part is that through our collaboration at UC San Diego, we have the ability to do magnetic recording, so magnetoencephalography, MEG, with one of the highest resolution systems in the world. And so here's what we're going to have, what we have people looking at. And it's terribly boring, but this is what happens. So you look directly at the cross, and then the little dot moves. And so we see in one hemifield, again in one hemifield, now across that midline. And so we're going to be comparing the ones that are all on one side to then across the midline. And so here's what we're talking about. So in a thing moving within the MA field, it's going to be a very short distance in the brain. It's going to be just a little bit between here. But to get across the midline, we're going to have to measure these tracks that go all the way across. And so there's the speed limit. This is the highway that the refraction ratio will depend on. So the speed of processing is super, super important. And if you'll indulge me, I'll give the little demo that I love on the speed of processing in the visual cortex. So I want you to hold your hand in front of out in front, and shake your head. Nice, and the hand stays nice and clear. And that's because your eyes are moving very quickly and keeping the visual world stable. Now what I want you to do is keep your head still and shake your hand. And what you see is that there's a speed limit. You can't see, your visual system can't track it fast enough. We have the biggest computer in the world between your ears, and yet it can't do this tracking. And the speed limit is probably what's going on here. So in other words, by putting all those pieces together across the visual fields in the knit together visual consciousness, we have to do, there are limits to that, and that's the speed limit that we're looking for here. And this, I think, is the reason, and what we're going to be looking for is this highway across the midline from one side to the other. So as I said, we have exceptional colleagues who measure the magnetic fields of the brain. Interestingly, as Stuart probably knows, they're most likely to be coming from the pyramidal cells, which are very interesting from a consciousness point of view. And being able to turn those on and off, like in our anesthesia experiments we've heard about already, is going to be a subsequent experiment that we do. And so what we'll be seeing is, as you see in this other side of the magnetic field superimposition over the anatomy. So we'll be measuring the time over the distance of connected areas. So we need extremely precise tractology. We're measuring the tracts of the brain, and we'll be using the diffusion tensor imaging MRI for that. We have the magnetoencephalography, MEG, to look for the sources and how they're linked, and then we'll be looking at how they connect together. So there are a few issues on this experiment. Curiously, there's something called nasal bihemispheric innervation. In other words, turns out that the brain is a little bit cute, and it sends images from one side and the other. So we're going to need to optimize the stimulus for our experiment, and then we'll be thinking about links in other regions. So I just wanted to say thanks to all of you for paying attention, particularly to our sponsor, the Herman P. and Sophia Taubman Foundation, and please email Gabe or myself at UCSG. Thanks very much.
Yeah, you're good to go.
All right, great. Hi, everyone. So sad I can't be there with you right now, but excited to present this work and close out this concurrent with fascinating talks so far. So I resonate with the last presenters, where I'm also interested in kind of the mechanisms of which underlie a lot of the processes that occur in the brain, specifically the more abstract, such as meta-awareness. And so I'll be presenting kind of work in this vein, only not so much at the cellular level, kind of the really broad abstract, what are the general mechanisms going on, specifically in mind-wandering paradigms. And just a quick note, this is version number 2. So it's slightly different than the abstract that I submitted. And I guess just as an introduction as well, my name is Isaac, and I work with Michael Graziano, and we study the attention schema theory. Okay, so the broad question that I'm interested in is, why does the mind wander? And the mind wanders for many reasons, but I'm specifically interested in this one specific instance, which is during focused attention meditation. And in focused attention meditation, we can think of it consisting of 2 tasks. The first task is to attend to your breath. Maintain attention on breath for the entire duration of the time in which you're meditating. And then the second task is to pay attention to where your attention is. This is sort of metacognitive monitoring procedure that is occurring simultaneously in addition to the task of attending to breath. And I don't know how many of you have meditated in the audience, but meditating is freaking hard. And it's not hard because focusing on one object is hard. We can all do that. But it's hard because focusing on an object for an extended period of time, an extended duration, is challenging. Particularly when that object you're trying to attend to, the breath, is rather uninteresting. And there are more fun, exciting things to think about, like this cute cat that for some reason is munching on his paw. And so my meditation therefore produces these episodes of mind wandering or attentional lapses, where there's this unintentional shift away from a target. There's this period of awareness, you know that you're attending to breath, and then there's a shift towards something else, this period of unawareness. And so one theory puts forward that the reason for these attentional shifts, unintentional shifts away from a target is due to a failure of control. And we can think of cognitive control as a method for manipulating behavior to achieve some end, particularly ends beyond simple reinforcement, so beyond just stimulus response. And just to kind of give a more concrete example, control is classically illustrated in the Stroop task. And in this task, participants are presented with a word such as this, and there are two sorts of responses they can make. The first is the automatic response, so to respond with the word, in this case, red. And this is the automatic response because we read words all the time, right? It's very well trained. We've been doing it since we learned to read. And the second is the controlled response, which is to respond with the color of the word. And we are entirely capable of doing this, responding with the word's color, but it's not something we typically do. So it's not that control is necessary to override the automatic response in service of a different non-conditioned goal. And so recently this idea of automaticity versus control has been extended to continuous states. So in the mind wandering paradigm, the automatic default state to be in is to mind wander, or this an explorative procedure seeking to maximize the value of what is on mind in the absence of an explicit goal. And then the controlled response is to pay attention to breath, to meditate. And it's thought that a top-down frontal parietal network is responsible for maintaining these on-task states. And in order to do this, there are 4 control functions of which it executes to maintain these on-task states, 2 of which I'm going to be talking about and further describing today. The first is applying activation. So maintaining that the breath is an important or salient thing to pay attention to throughout the duration of the task. And furthermore, we can kind of think of this function as an attentional control signal that biases information to produce controlled behavior related to the task. So this takes the form of higher versus lower control signals, where higher control signals result in a more vivid representation of the breath in mind, whereas a lesser signal intensity is going to result in a less vivid representation. The second function of which I'm going to be talking about is this metacognitive monitoring procedure, which corresponds to checks or samples of what is on mind. So, for example, I can ask you right now, what is it that you're thinking about? To which hopefully you would respond, I'm thinking about exactly what the presenter is talking about right now. And this sort of function is different, disassociable from other monitoring mechanisms that occur in the absence of meta-awareness. So when you're driving your car, you're likely monitoring the road, but you're not explicitly aware of the actions you're doing to complete the task of driving your car. So this is the metacognitive introspective version of monitoring that is executed by this top-down frontal parietal network. So what I'm going to propose today is a model that sheds more light on the mechanisms behind an attentional lapse during focused attention meditation. And we're going to specifically suggest that these shifts occur in the absence of a metacognitive monitoring procedure to sample internal contents and then implement the appropriate control signal. This is going to produce a drift away from the task of meditating towards other mind-wandering procedures like cute cats. I'm going to use a control theoretic model to build this idea of unintentional mind wandering out. And I'm going to speculate as to how this model could produce differences in individual ability in meditation. Okay, so I mentioned I'll be using a control theoretic model. I'm not going to go too deep into this, but any sort of sort of model like this, a closed loop control theory model, is going to have 3 components. The first, a dynamic system. The second, a controller which acts on the dynamic system. And then third, a feedback aspect of which observes how the effect that the actions have on the dynamic system, the states, and then determines how to best apply a next action on the next time step, given how the previous action affected the state. Okay, so I'm essentially going to take that framework and apply it to what could potentially be going on during meditation. So on the x-axis, we have time here, and on the y-axis, we have S of t, which corresponds to your current state of attention, what is on mind. Higher values of S of t then are going to correspond to a more on-task state, and lower values a drift towards mind wandering, more off-task. At the beginning of the task at the onset of meditation, as well as in periods following a button press, you're going to be in a state of near optimal performance, or the desired states. Over time, though, you are going to drift towards mind wandering, this equilibrium of which the mind naturally evolves to over time, given no control. So then with the addition of control, you can maintain on-task states. So we're going to basically say that how much control then to apply on a given time step is going to depend on the error. And the general idea here is when you're on task at the onset of meditation, task performance is high. There's very little error generated. So as a result, a low amount of control is going to be applied. Conversely, as you get closer to mind wandering, there's going to be a very high error generated because your attention is not where it's supposed to be. And as a result, a large control signal is going to be generated. And this idea is based in the expected value of control theory, which is essentially saying it's more valuable to apply control when super off task compared to applying just a little bit more attention when task performance is already sufficient. And this relationship is going to take the form of a scaled sigmoid, where there's a maximum control signal to exert as well as a minimum control signal. And now with the addition of control, instead of drifting to mind wandering, we're going to drift to a more stable state. And we're going to call this the mean on-task equilibrium. And this corresponds to the most common phenomenological state during meditation, one where you're on task and working hard to maintain this on-task state. And then we're going to implement the metacognitive monitoring procedure in order to get at these mind-wandering events. And metacognitive monitoring has previously been described as this intermittent procedure, but the details of how it works has been left largely unspecified. So we're going to attempt to provide kind of a more rigorous definition to that, saying that metacognitive monitoring, or internally sampling and checking what is on mind, is constrained by this probability function. And similar to the control, in a high on-task performance state is going to correspond to a high probability of checking or knowing what is on mind. And intuitively, we can think about this as at the beginning of meditation, there's a high degree of confidence, right, that you know where your attention is. However, as the task continues and you get closer and closer to mind wandering, there's less of a chance that you're going to check or sample what is on mind. And we're going to propose a specific relationship here between monitoring and control, being that if you fail to check what is on mind, you're not going to apply the appropriate control signal on the next time step. And this relationship is also going to take the form of a scaled sigmoid, where there's a maximum probability as well as a minimum. And the minimum is set to a non-zero value, as even when you are completely mind wandering, there is still kind of a recursive check that can happen. There's still a probability that you're going to sample even if you're mind wandering. So that's why it's a non-zero value. Okay, so now with both of these functions, we can simulate kind of the meditative process as we're conceiving it. So at the beginning, attention at the beginning of the meditation task, attention to breath is near optimal, and the probability of sampling is high. There's a good chance that you know what is on mind. Then we're going to drift to a more stable state, one that is a balance of optimal control, a compromise of optimal control and appropriate task performance. Then some time step is going to occur where you do not check or sample what is on mind. As a result, this is going to cause the state to further drift towards mind wandering and simultaneously decrease the chances that you sample on the next time step. And multiple time steps then without sampling, we're going to define as mind wandering, or 3 standard deviations below the mean on-task performance. And again, like intuitively, you can think this to be somewhat true. As you're meditating, you're constantly, judging how on task you are. And anything that you determine to be significantly different than, your common stable on-task performance, is going to be what you would consider a mind-wandering event. And we're going to define that period as the period where you recognize that you are mind-wandering as this recognition of mind-wandering, that an intentional lapse has occurred, which corresponds to the button press, when subjects might press the button if they're told to press a button when they notice their mind to have wandered. Following this recognition, attention is going to reorient back to the breath from this large control signal, which signifies a large error. And then following the reorientation back to the task, attention to breath is going to decay back to a more stable state. Okay, and so then we can simulate this over an arbitrary amount of time. So in this case, on the x-axis, we have 300 seconds or 5 minutes. And just to recap, so you can see throughout this 300 second duration here, most of the time you're on task, but there are these periods of lapsing and reorienting attention back to the target in the model. And just to kind of recap what produced this data are just two functions. The first is an attentional booster control function, which takes in the error and produces and says how much control should I exert. And the second, a metacognitive monitoring procedure that checks or samples the internal state. Okay, so this model is cool and all, at least I like to think so. But the question is, does it relate to anything in the real world? Always a poignant thing to ask oneself when doing these sorts of models. And so it's inherently hard to get at the neural machinery. But I just attempted to look at behavioral data to see if I could simulate if this model matched up to behavioral data. So in order to do this, I had 22 participants do 2 5-minute meditation runs, and they were told to press a button if they noticed their attention to no longer be on the breath. And in this graph here, you can see button press count, as well as the interval between button presses. And if we were to look across the group data, participants pressed the button a mean of 11 times. And just to recap again, this is the average of 2 runs per subject. That's why you have half button presses. And the mean interval between button presses was 37 seconds, although there is a huge variation in the amount of time between button presses. Okay, so then I essentially simulated 22 subjects. And I said that the period of which there was a sample on the previous time step, but before that there was not a sample, and you're in a state of mind wandering. That is what this button press was according to the model. I essentially summed those and looked at the intervals between these simulated button presses. And then I compared that to the behavioral data that I had. And just to be clear, I didn't do any sort of rigorous optimization here, which is something I'm looking into currently. All I did was manipulate the parameters of the metacognitive monitoring function as well as the control function to match the means and variances of the behavioral data. And so according to this set of parameter values, you can see that it does a very good job of matching subjects that are closer to the mean here, as well as the interval, so mean button press as well as interval mean. But it does a terrible job, or not as good of a job, for accounting for the outlier cases. So subject 09 press the button once, or subject 21 press the button 22 times. And while other studies would just kind of throw away this data and say that is not of interest, something was going on here, I wanted to include this data because there's still valid data points, and we still want to figure out what's going on here, granted that they still, gave some sort of attempt at the task. And so there's essentially 2 ways in which our model would explain why participants would press the button very few number of times, corresponding to subjects like these. The first explanation is subjects are just really good at monitoring. Specifically, they're always aware of where their attention is, and as a result, they're not letting their mind to drift towards mind wandering, which corresponds to a shift of these parameters of the metacognitive monitoring procedure to the left. The second explanation is that participants don't really care. Unfortunately, they didn't try too hard, so they weren't applying control so often throughout the entire task. Obviously not an ideal situation, but definitely something that could have happened. And essentially they allow more error to occur on the task before applying control signals. So this would be less motivated subjects potentially. And we can do the same thing to explain a higher number of button presses. So for subjects like these, that being that they're really bad at monitoring. So they're not checking what is on mind. Attention is constantly kind of evolving to mind wandering. As a result, a bunch of button presses. Or subjects just really care about doing well in the task. So any small deviation in attention to breath is going to produce a trigger, a button press. So very little error is allowed before applying larger control signals. Okay, so those are the behavioral predictions, but one really cool thing about, this space is it's pretty hard to define a lot of theoretically, it's harder to more rigorously define a lot of the theoretical predictions that people have, but with our model, we can take a stab at that. So one thing we can test is how much time do participants spend on task in a 5-minute meditation run by saying that anything on task is 3 standard deviations below the mean on task performance. Anything above that is on task. And so our model reports that across this 5-minute duration, across all subjects, they're mind wandering according to attentional lapse at a rate of about 10%. The second thing we can do is just isolate specifically the metacognitive monitoring procedure. So how often is it that participants are checking or sampling what is on mind? And from here, we can see that 75% of the 5-minute run, S of T, was sampled. And then lastly, we can also look at how much time is spent mind wandering prior to a button press. And just from behavioral reports, this period is noted to be quick. Subjects kind of quickly ascertain that attention is not where it's supposed to be, and they can reorient attention back to breath fairly easily. So this was more of kind of a sanity check, being that on average there is 2 time steps, 2 seconds across 22 simulations prior to button press where mind wandering occurred. Okay, so now just to kind of recap everything, I began by asking the broad question, why does the mind wander? And specifically look to answer that question in a more refined paradigm, focused attention meditation, where these unintentional shifts of attention occur from on-task to an off-task state. I proposed that these attentional lapses during meditation are a result of first, a metacognitive monitoring procedure to sample internal thought contents, and then apply the appropriate control signal to breath. I then defined this more rigorously in a control theoretic framework, and then sought to convince you at potential reasons according to the model of why this could be the case in different individuals. And then lastly, I also have fMRI data, which is an extension of this study. Subjects were scanned while they were performing this experiment. So that is sort of a next direction here, as I want to see if I can look at the metacognitive monitoring procedure and as well as the control function, and see if there are areas in the brain that map onto the extracted activations from these different nodes in the model. And I just want to thank all my collaborators and all of you for listening.
Okay, that's a wrap. Thanks, everybody.